Welcome to this presentation. My name is Peter Ankomah. Tagged-ICP is an iterative closest point algorithm that uses metadata knowledge for improved matching of 3D protein structures. It is based on the well-known ICP algorithm, which is extensively researched 3D registration algorithm because of its simplicity and modular nature. So ICP aims to best align two-point clouds by minimizing the distance between corresponding points. The algorithm iteratively computes correspondences and distances and applies a transformation that minimizes this distance. The algorithm works well without requiring knowledge of additional features of the point cloud, except the geometry of the shapes. Preprocessing of data is also not required for the registration process, and the methods can be implemented with different existing techniques. So for instance, the closest point search method can be implemented differently using parallel processing, metadata information, or registering the subsets of the point clouds. The ICP algorithm iteratively computes the transformation that aligns two point clouds. This algorithm shows two point clouds P and X iterated over K max, which is the maximum number of iterations set for the registration process. Then a transformation is computed that aligns the data point cloud to the corresponding closest point in order to reduce the distance between the data point cloud and the model point cloud. And these above computations are repeated until the change in mean square error between the current and previous iteration is less than the set threshold. Even though ICP is well known and is a standard shape registration algorithm, it suffers from some limitations, such as the need for a good initial transformation. Now, this requires a user to normally use a smaller optimal transformation to ensure good registration results. The complexity of the point clouds have also been noted as affecting the quality of registration, and in some cases, whether the algorithm converges or not. The algorithms actually perform better when the shapes are similar, and the registration process converges with a smaller initial transformation. And large point clouds have also been noted to slow down the algorithm due to the number of correspondence searches to be made at each iteration. In fact, in Donoso's 2017 article, they noted that registration results were better in some datasets than others. The research studied the registration of point clouds using scenes from a mining environment, and according to the results, most ICP algorithms were shown to perform poorly with this terrain data. The research compared 73,728 ICP variants and found none to be accurate, precise, and fast at the same time for map terrain data. This is because specific geometric information which would have benefited registration with this dataset from terrain maps are not used in the matching process. But also, for each domain, specific datasets, there should be a careful selection of techniques and strategies for each stage of the ICP process. So even though ICP performs well, it can not only convert to a good match, where the algorithm finally finds the real correspondence for each point, and the visual overlap looks acceptable, but it can also convert to a bad match, where the distance between the real corresponding points are huge. In some instances, ICP will not converge at all, and in some of our experiments using 3D protein data, the data shape has been found to fly away indefinitely. So this brings us to our tagged ICP, which is our improved variant of ICP for matching the 3D structure of proteins. Now, proteins are usually matched with their sequence or structure or, both information. However, matching with the 3D structure has become more important because of how it is less sensitive to changes such as mutations compared to the sequence information. Well, currently, ICP is already used to match the 3D structure of proteins, to identify similarities, to study common ancestry, and related protein functions, among others. Tagged ICP uses the point's own atom type, and the atom types of the k nearest neighbors are ordered to find a correspondence for a point. The algorithm partitions the search space for correspondence by the atom type. To find the correspondence for each atom in the data point cloud, the search considers only atoms of the same type, having the same neighborhood information. Depending on the distribution of molecules in the point cloud in terms of the size of the atom partitions, the speed up gained in the search for correspondence in a smaller set space reduces the time to convergence of the algorithm. Tagged ICP only modifies the closest point search method of the base ICP algorithm, and this modification uses the metadata of the protein shape to speed up the search for correspondence and improve the registration accuracy. The model point cloud X has been partitioned by the tag P, and only the partition corresponding to this tag of P is searched. A protein molecule is an organic molecule consisting of typically thousands of atoms, mostly carbon, hydrogen, and oxygen. Now, this image shows the 3D structure of a protein molecule loaded from a protein data bank file, PDB file. This file was obtained from rcsb.org, which is a repository of PDB files. Cell Unity is a third-party application that is used to read the contents of the PDB file, and this application automatically generates spheres representing the atoms in the particular PDB file. Cell Unity represents all those atoms in the file, and this yields the 3D structure of the protein that we can see on the screen right now. The left-hand pair of structure represents the protein 3D structure with only the individual atoms scaled down, which is the point cloud to be used for the matching, while the right-hand structure represents the original scale of the molecule loaded in from the PDB file. Now we scale down the 3D structure in order to be able to easily analyze the overlap of the regions after the matching, especially for regions at the middle of certain sections of the molecule. It is difficult to visually inspect the overlap when using the original scale shown in the right pair of image. The experiment generates two types of graphs, the match quality and the convergence graphs. The match quality graph is a cumulative histogram showing what percentage of points have alignment errors smaller than the value on the x-axis. So a good match would mean this graph goes up very quickly. The match quality graphs represent the quality of the registration based on the percentage of point pairs and the distance between them. This graph shows the quality of the match after the shape matching process is complete. You can see it as a statistical equivalent of viewing the overlap of the shapes after the registration process is complete. These two graphs represent the match quality results for matching same shapes at 30 degrees and 19 degrees rotation of the data point cloud along its x-axis. We can clearly see from the graphs that both Tagged-ICP and ICP had a perfect match. The horizontal axis range is an exponent of -7, which is in orders of magnitude smaller than, for example, the distances between atoms in a molecule. This experiment represents a simulation of noise added to the data points to mimic noise added to the point clouds by a position measuring device. The noise vector, represented by 0.001 to 0.05, represents the extent to which an individual point position is moved in the radius of the point cloud. So noise value 0.001 represents a maximum of 0.1% shift, and 0.05 represents a 5% shift in the position of the molecule. As you can see in the graph, for stronger noise levels between 3 to 5%, Tagged-ICP is clearly better than original ICP because it shows a quicker rise than ICP. These two graphs show the convergence of ICP and Tagged-ICP at different initial rotation angles and iteration. We notice that Tagged-ICP converges faster at all angles of rotation, and this is due to the use of the metadata or the tag to help with the matching. ICP 180 displays the known limitation of ICP, that it can diverge if the initial rotation is too large. However, Tagged-ICP 180 managed to recover from that. Tagged-ICP converges earlier at fewer iterations, but possibly at a higher mean square error. This is because ICP considers any two atoms that are close together as a good match. Tagged-ICP forces atoms to match with the same tag. So with the introduction of some level of noise, Tagged-ICP might revert to the closest atom of the same type, that is within the same atom type partition. So as the level of noise increases, Tagged-ICP might be worse in terms of distance, but it's better in terms of matching atoms to the same tag. Proteins are made up of amino acid sequences, and mutations wrongfully alter these sequences, such as wrongful additions and deletions to these amino acid sequence characters. This graph shows shape registration of two atoms, one being a mutation of the other. The quality of match graph shows that both algorithms had a similar performance in terms of their percentage of point pairs above an alignment error. However, Tagged-ICP converged at a higher mean square error than ICP. Tagged-ICP considers same atom types and matching neighbors when finding a correspondence for a point. However, when this match is not found, the algorithm reverts to the closest points amongst the same type of atom. So Tagged-ICP might be worse in terms of distance, but it's better in terms of matching atoms to the same tag. So this slide shows an animation in Unity 3D of registration using ICP and Tagged-ICP of the same point cloud. Our experiments show that using additional metadata information of points in the shape matching process can increase the accuracy of the match as well as result in faster convergence. Because the closest point search method uses the atom type and k nearest neighbors to ensure a good match, the process becomes meaningful, and this also reduces the corresponding search space, effectively making the algorithm converge faster. For our ongoing and future work, we are extending ICP with matching subsets of points. We are also looking into registering partially overlapping molecules as well as extending the registration of overlapping molecules with confidences. Thank you very much, and I'm happy to answer your questions.